abscisic acid and ethylene are growth inhibitors, but ethylene are the group in case of fruit ripening. So fruit ripening, it is a one kind of promotion of growth. So as for their function in the fruit ripening, they have been also been referred in the growth promotion, and in case of inhibition they also been growth inhibited. At the excess concentration, the excess fruit ripening that leads to the division of the inhibition of the growth, that also been called. In the next system, the first hormone that we are going to study, that is auxin. So auxins are nothing but these are the first class of plant hormone that to be identified. Means this is the first hormone that is identified as a plant growth promotion. The term auxin, it is derived from the Greek word auxine, that is been referred as a auxin, which means to grow. So for that reason, the auxin has been coming from the word auxine, that is a Greek word, and when we translate or take the word, that is the etymology that we can take for the auxin, that is a auxine. Auxine and auxine, that are the two. Auxine, the different pronunciation, that means to grow. So all those growth regulating organic compounds which are produced at the tip of the roots and the tip of the stem as a result of metabolism and transported to the region of elongation, causing elongation of cells called auxin. Means these organic compounds which are produced at the tip of the stem or at the tip of the root, means at the initial position, so that the tip consisting, that we can call as a meristematic zone, and these meristematic zones are capable to form these conditions, these types, so that are been known as auxins. Then they are responsible for promotion or the elongation of region where they are coming in the stem and they're transported to the lower region, that is called as a basipetal succession, the basipetal movement towards the base. So and when they are coming towards the base, at that time, that is movement is and transportation is called, and that transportation is responsible for the elongation of cells. So that is nothing but it is called as a auxin. Means any part that has been grown by that particular organic components, means they are the auxins. Both in a natural and synthetic auxins are known, and all have similar effect on the plant growth and development. Means when we study about the auxin, in this case, these hormones are being classified into the natural and synthetic ones. In case of, when you take the example of indole acetic acid and indole butyric acid, these are coming into the natural ones or the natural auxins that are synthesized by the plant system in its metabolism, or organic components that are synthesized by plant system in case of indole acetic acid and indole butyric acid. But when you go through the synthetic auxins, that is naphthalenic acetic acid, and in case of 2,4-D, that is 2,4,6-dichlorophenoxyacetic acid, that is dichlorophenoxyacetic acid, 2,4-D. So it is also been one kind of auxin, but that was the synthetic one. In case of indole acetic acid, indole butyric acid, these are the natural ones, and in case of naphthalenic acetic acid and 2,4-D, these are coming on the synthetic ones. But in case of natural and the synthetic ones, they both have similar effect on the growth. Means there will be no any differentiation in the action of growth. There is no any differentiation in the effect of the growth or the physiological effect in case of. So in case of the discovery of the auxin is Thiman in 1948 defined auxin as organic substances which promotes the growth along the longitudinal axis when applied in a low concentration to shoots of plants as far possible from their own inherent growth promoting substances. Means they require Thiban defined the auxin as organic components Means basically these are organic components consisting carbon and hydrogen group. So they are been as organic components or the organic substances which promote the growth in a longitudinal axis. Means the growth has been promoted in a longitudinal axis. Those components, those organic components that are responsible to growth in a longitudinal axis and in a low concentration. So these two conditions has been proposed by Thiemann in the definition of Thiemann. And the third condition is that these are inherent and growth promoting substances. Means plant are inheriting capability to promote their substances, to synthesize that substances for its growth. Auxin passes from shoot tip to the region of elongation. See, in case of when the growth happens, the initiation, elongation and the differentiation. So from the initiation part, it has been passes away, it has been transported away to the zone of elongation or the region of elongation in the movement that is the basipetal from apex to base, and in stem in acropetal from base to apex in root. Means in case of shoot system, it is a basipetal movement. Means some components, some organic components or auxins, they have movement from its zone of initiation to zone of elongation. Means from apex to base, that is called basipetal movement in case of stem. But in case of root, it is base to apex. That is called acropetal succession. When it is towards apex, it is known as acropetal. When it is towards the base, it is called basipetal succession. Succession or the movement that you can call. See, now the concentration has been here been represented in case of auxin helps in the elongation of both shoots and roots. However, optimum for those the two is different, quite different. That is 10 ppm. 10 ppm means it is a parts per million. Means 1 million is equals to 10 lakhs. Means in a 10 lakh ml concentration, that is only 10% of that particular auxin. It is a very little bit concentration. Means 10 ppm for stem and 0.0001 ppm for root. Means it require in a very low concentration for the promotion of the growth, for its action in a very low concentration. When in case of the shoot concentration, it is a 10 ppm. Means should require more concentration than the root for the, and the auxin biosynthesis occurs in the shoot apices, leaf primordia, and developing seed from amino acid, and developing seed from the amino acid tryptophan in presence of zinc ion concentration. Means the auxin biosynthesis occurs in shoot apices where it synthesized, rather biosynthesized, or it is naturally synthesized, though it has been referred as a biosynthesis. In case of shoot apices, the region where it synthesizes shoot apice, leaf primordia, the initiation of leaf or the initiation point of the leaf, that is called leaf primordia, and in the developing seed. So in case of shoot apices, these are the meristematic zone. In case of leaf primordial, these are the lateral zones, and from the seed. But in case of seed, there are some conditions. Means in case of amino acid, the tryptophan is responsible to synthesize the auxin. So that is called the zinc plus ion. In the presence of zinc plus ion, the tryptophan synthesizes the auxin in the developing seed. And most of the other members of auxin family, that is indolacetic acid, which generate the majority of auxin effect intact plant and is the most potent native auxins. See, as I told in the last slide, that is the indolacetic acid is the major auxin, and it have the in the intact plant system, it is the major auxin and have most native auxin. Means in the auxin family, it means there are number of representatives of auxin. In the auxin family, 1 naphthalene acetic acid, indolacetic acid, these are the families of auxin. There are number of organic components or the auxin components. But in case of in rest of that, the indolacetic acid, it is the majority of auxin that have effect in the intact plant, and it is a most potent native auxin. And as the native auxin, now see what is the indole acetic acid the characteristic. Uh, in case of auxin, its stability is controlled in many ways. Why it has been called as a native auxin? Indole acetic acid, why it has been called as a basic native auxin? Because its stability is controlled in many ways in plants, from synthesis, conjugation to degradation of molecules. So there are three different characteristics has been given. First one, its synthesis, means it is biosynthetically synthesized or biosynthesis. Second, its conjugation, means it is connected, conjugated with the parts that are been for the growth, for in case of zone of elongation. It is conjugated, means it is mixed. And then after the conjugation, there is a degradation, means it's degraded and the growth of the plant took place. Means three major characteristics had been given, so they had been referred as a native auxin in a, and that requirement in the situations and the different situation. See, the natural occurring, that is called endogenous, naturally occurring, it is a endogenous auxin in plant include indole 3 acetic acid, 4 chloroindole 3 acetic acid, phenylacetic acid, indole 3 butyric acid, byric acid, and indole 3 propionic acid. So these are the major auxins that are naturally biosynthetically produced in the plant, synthesized in the plant. And the synthetic auxin include one naphthalene acetic acid, 2,4-D 2,4-dichlorophenoxyacetic acid. So these are the two. See the structure of auxin, and the carbon and the hydrogen is present. So they are been referred as the organic components. See the structure of auxin. So I think so. This much is sufficient. In the next lecture, we will go through the discovery of auxin. So I think so. This lecture has been beneficial for the students that are going to introduction of plant growth regulators. In the next lecture, we will move towards the discovery of auxin and the action of auxin. Thank you. Thank you very much.